Hi, my name is Carolina Herrera, and this is my presentation on differential genomics and transcriptomics between tyrosine kinase inhibitor sensitive and resistant BCR-ABL dependent chronic myeloid leukemia for the class Cancer Biology at Texas A&M University, Kingsville. Chronic myeloid leukemia, abbreviated as CML, is a cancer of the bone marrow, and it happens when there's high counts of white blood cells, and it's caused by a genetic defect in the ABL-BCR gene. The main difference between this form and acute forms of leukemia is that this form progresses at a slower rate. The materials for this research are CML patients, 4 healthy patients, molecular inversion probe-based array, and the human transcriptome array 2.0. Before I start in the abstract, these are some terms that will help you have a better understanding throughout the presentation. Tyrosine kinase inhibitors are a pharmaceutical drug that inhibits tyrosine kinases. Molecular inversion probe-based array is a process where one captures and enriches specific regions of the genome. And the human transcriptome array 2.0 accurately detects all known transcript isoforms produced by a gene. Now for the abstract. It's known that BCR and ABL fusion protein is enough to induce chronic myeloid leukemia, but additional genomic changes are required for the disease to progress. Because of this, they profiled control and tyrosine kinase inhibitors alone or in combination with other drug-treated CML samples in different phases. And they were categorized as drug-sensitive and drug-resistant. And this was off the basis of the BCR-ABL transcripts. Imatinib is a form of TKI and has been shown to work if the BCR-ABL transcripts are less than 10% after 3 months, less than 1% after 6 months, or undetectable after 18 months. But in treated patients where BCR-ABL isn't detectable, the chronic myeloid cancer moves to the advanced phase, or they show remission. And because of this, genome-wide profiling of different phases of imatinib-treated chronic myeloid leukemia is expected to uncover signaling pathways and molecular mechanisms involved in imatinib treatment at different phases of CML. Genome-wide scoring of SNMPs in different phases of imatinib-treated CML will also further help us understand the resistance mechanisms to TKIs. The array that was used in this research measures a total of 67,528 genes. Out of the total, only 2,073 genes were differentially expressed, 1,425 of these genes being coding genes and 648 being non-coding. Compared to control among all CML samples, 69 genes were upregulated, 49 being coding genes, 20 being non-coding, and 2,004 genes were downregulated, 1,376 being coding and 628 being non-coding. What this shows is a distinct clustering of the 35 chronic myeloid leukemia samples and the 4 normal controls. And this is shown in the figure A, which is the left figure on this slide. When comparing clusters, samples with undetected copies of BCR-ABL were classified under the first cluster as non-sensitive cases, which is cluster 1, subcluster 1. shown in this figure. In the second cluster, subcluster 1, it included all control samples. And in the subcluster 2, it included the chronic phase, new cases, and cases where copies of BCR-ABL were undetectable. The cluster 3 included samples showing more than or equal to 1 to 10% copies of BCR-ABL, and this is shown in the second to last column of this figure. Copy number variation, which is CNV abbreviated. Copy number variation profiling were done for 39 chronic myeloid leukemia samples, 5 of them being control samples, and they were categorized on the basis of CNV's percent loss of heterogosity and the percent change, which is all shown in this figure. In conclusion, the genome abnormalities contribute to the transformation from chronic myeloid leukemia to chronic myeloid leukemia blast crisis. Genes included in the pathways FOS, TGFB, RZ, TPX2 can be used as markers for chronic myeloid leukemia development. And genes included in the pathways LAPTM4B, HLTF, CFH, PIEZO2, ANGPT1, CD109 can be used as markers for chronic myeloid leukemia resistance to therapy. And here's my cited source. And that's it for my presentation. Thank you very much for your time.